Patrick, and I'm an intern on the Foundry's communication team. Today, we'll be interviewing Dr. Bruce Cohen, a staff scientist at the Molecular Foundry, whose work combines chemistry, biology, and nanotechnology. Could you tell us a little bit about yourself, Bruce?
Sure. Thanks, Yushi. Hi. Yeah, Bruce Cohen. I'm a scientist at the Molecular Foundry. I do research in the area of, well, nanoscience, obviously, but chemistry and biology. So I have a research group that's interested in developing new nanoparticles and optical probes that help us understand how cells work. My background, I originally got a PhD in chemistry at UC Berkeley, doing some synthetic chemistry and also biophysics. And then I went and did a postdoc at UCSF with Lily Jan. And a real sort of change. It was biology lab, a lot of geneticists, cell biologists, trying to look and see what we can do as chemists to help understand biological problems. And then I made my way to the Foundry, where I've been as a PI ever since.
Thank you for sharing, Bruce. So, to begin our questions, could you give us a brief description of your research group?
Sure. So a little more specific to what I said. We are interested in harnessing nanoscience. So there are a lot of nanoparticles that have very unusual and interesting optical properties. And one of the things you can do with materials that have optical properties is biological imaging. So you can study the precise molecular workings of cells and groups of cells. And we're very much interested in taking these new materials, which are often developed sort of without biological applications in mind, and making them biocompatible and figuring out how to make them go where they need to go in a cell or in a group of cells, and then developing imaging methods to try to better probe the molecular workings of cells.
Thank you so much.
Sure.
Going back, why did you choose this field of study? Is there a particular moment that sparked your interest?
Yeah, so I was working as a chemist and seeing um, people start to migrate towards more serious biology, and a lot of that initially was is in the field called chemical biology, was in the field of immunology, of developing new chemical approaches to immunology, and looking a little more closely, I realized that neuroscience was a pretty large untapped field for chemistry, and neuroscience has a lot of chemistry in it. We have neurotransmitters that are small molecules, and we have peptides and neuropeptides, things like that. And so the idea of doing chemistry that is useful for an application rather than just doing chemistry for chemistry's sake was always appealing to me. And I thought I could have a much larger impact going into a field where there wasn't really a lot of people working as chemists. And so I went to UCSF and I worked in a biology lab. And one of the things I noticed almost right away, and I had sort of, when I went, I sort of had these ideas of things that I would do. But when I went to UCSF and I saw a lot of my friends working in lab, the chemistry they would use every day was fluorescence. They would be looking in a microscope, tracking things around under the microscope, identifying proteins and channels that they were studying. They would use fluorescence for studying their DNA, their gels, or sort of a lot of standard reagents. And it occurred to me that this was a pretty fertile ground for introducing some novel chemistry to help understand neuroscience. And and that worked well. I did some of that while I was a postdoc. And as that was happening, I saw at Berkeley and other places a new class of optical probes, nanoparticles. So these were quantum dots that people were starting to publish and talk about. And it seemed like a really exciting area to, instead of making organic small molecule probes, to make nanoparticles that had these really unique properties. And that's why I made the jump from what I was doing to more nanoscience in developing nanoparticles as optical probes for imaging. And that's sort of where I've been at the Foundry ever since.
Thank you so much for sharing, Bruce. Diving deeper into your work, what would you say is the biggest challenge of using nanoparticles for bioimaging?
Yeah, the biggest challenge is that they're not particularly, when we make them initially, they're not particularly biocompatible. They're not even in water, right? So we have to do all of these things to get them to go into water. And then once they're in water, to get them to retain the optical properties that we want and to get them to not clump together. So they'll oftentimes will aggregate and just sink to the bottom. That's a problem that the field has had for a while, and that's not unique to us. One of the things, the bigger problem I think that we've been addressing is how to get them to go to the right place. So cells are immensely complex things. We have like thousands and thousands of different proteins at any given time. If you want to study things on the surface of the cell, like we do, like receptors or channels or things like that, there are hundreds of thousands of different types of those on any given cell. And then the thing that makes it most confounding is all of those proteins, the channels, the receptors, the enzymes, they're all composed of the same things. They're all composed of the same 20 amino acids. So trying to address an optical probe, a nanoparticle to the right position in a cell is very challenging. And fortunately, nature has come up with a solution for this. So nature produces antibodies, we produce antibodies that can distinguish one protein from another with high specificity and get those our optical probes to the right place. But the biggest challenge, I would say, just to sum it up, is both biocompatibility of our optical probes, making them happy in water, and also getting them to the right part of the cell.
Thank you. You published a paper recently on something called lock coupling. Could you explain what that is?
Yeah, so this was one of the ways we've tried to address this issue of how to get nanoparticles to a very specific location, a very specific protein within a cell or within a group of cells. And lock coupling is simply a chemical reaction that permanently binds together two proteins that often will bind to each other transiently. And in this case, the goal was to take antibodies, which are these proteins we all make that can distinguish proteins from other proteins, and they get those permanently affixed to the nanoparticle surface. So one of the challenges, if we want to make a nanoparticle that has an antibody attached to it, there's two key challenges. One is how to make that specific thing, one nanoparticle, one antibody, and the antibody has to be in the right orientation so it can then also recognize those proteins on a cell that we want it to target. The second challenge there is to make that complex that I just described stable, how to make it stable. And the reason for that is if we have antibody on a nanoparticle and it can come off, which is sort of the typical way people do it, then once we put it in a cell, or if we're, say, working in an animal model and we inject it into a mouse, we have no idea if the antibody falls off the nanoparticle because the only thing we can see is the optical component is the nanoparticle. We can't actually see the antibody in real time. So we want to make one thing, one nanoparticle, one antibody, correct orientation, and we want to make it stable. And that was why we developed this chemistry called lock chemistry. And the trick there is to take a protein, a natural occurring bacterial protein that normally binds to antibodies, and put that on the surface of the nanoparticles in a very stable way. And once we do that, we can mix in the antibodies and they'll bind. But again, that's reversible. The antibody could come off. And so the lock coupling was an extra step we invented where we took this interaction and we made it permanent. What we did was we created a bond between the two proteins by adding a fairly simple chemical reagent. So that's what lock coupling is. Just add one reagent to proteins that are reversibly bound to each other and get them to be permanently bound to each other.
Thank you so much for sharing that aspect of your paper. Learning more, what are you hoping to use your paper's discovery for?
So we initially in the paper used it to image proteins on cancer cells. So proteins that are overexpressed on certain types of cancers. So these would be breast cancer and ovarian cancer. And there is a protein called HER2. So that was our first application, was to put antibodies onto our nanoparticles, lock them together, and then image that particular receptor in cells in a dish, and then even to image them in tumor models in mice. Ultimately, this collaboration with my colleagues at UCSF is to use this sort of technology to image cancer in patients undergoing surgery. And the goal there is to be able to see all the cancer cells. And the way it's done now is a surgeon, a cancer surgeon, will cut out a tumor largely by looking at it and feeling. So look and feel. And that obviously has its limitations. You can't see individual cells or feel individual cells. And so the practice in general is to cut out healthy tissue, or what appears to be healthy tissue, a certain distance around the tumor. And that keeps the helps keep the cancer from coming back. You ensure that you get all of it, but it has major problems. It can be disfiguring. You're cutting out healthy tissue. So our goal is to use our technology and combine it with some imaging technology that our UCSF colleagues are developing to be able to look in a tumor bed in real time and know exactly where those cancerous cells are. So using our imaging reagents that have antibodies against cancer markers and the imaging technology our colleagues are developing to be able to detect every cancer cell.
Thank you for sharing these applications of your paper. Now, learning more about you as a scientist, how do you keep yourself motivated when research poses obstacles or takes time?
So yeah, that's the trick, right? I think research always poses obstacles. It's if things were so straightforward that they worked the first time you did them, they're probably not very novel or interesting. If you want to work on, we really like to work on things that are difficult problems. And they don't always have difficult solutions, but things that are challenging. That's the sort of disruptive or groundbreaking or new things you can do if you're not doing experiments that you know will work in advance. And I think it's standard that when you're doing science to expect failure. That's kind of something that, when I have new students or postdocs, you have to kind of keep them up, it's a normal part of the process. You do an experiment, it doesn't necessarily work the way you expected the first time or maybe the second or third time, and you have to learn from those experimental failures as to what you need to change to make it to work. And it's a talent you develop too, I think. And some people are just naturally very good at this, and some people have to work at it. But I think most people who become successful scientists learn how to make the experiments work. And so, the question you ask about how do you keep it up, I think, it's a challenge, right? You view it as a challenge. Like, okay, how am I going to get this to work? What do I have to focus on? What have I learned from these experiments? And viewing it, I think, as an intellectual challenge makes it, that's a motivation in itself. And I think a lot of people, certainly like students and postdocs, respond to that. Like, how are we going to make this work? And they'll think about it, and coming up with a creative solution or something that nobody had thought of before, that's really kind of a reward. I think people enjoy that a lot.
Thank you so much for sharing. To close, is there any advice that you would give your younger self or a researcher starting in the field?
It's a good question. I think there's a lot of advice I'd give to my younger self, probably because science keeps changing and it's changed a lot, and the sort of criteria that people look at for science. Um, but what I would say generally is um, keep an open mind when you're doing experiments. And the reason I say that is a lot of our most interesting results with our nanoparticles and some of this bioconjugation chemistry are things we weren't planning. They're things we didn't expect. And so if you're kind of prepared for that to see an unusual result, and instead of writing it off as a failure in your lab, to say, oh, this might be interesting and follow up on it, that's been immensely successful for us. We've really, a lot of our most interesting discoveries, for me and my collaborators, have come from things we were not planning. So I would say keeping an open mind to your results, noticing some details that maybe weren't things that you were trying to do, but could be interesting if you followed up on them and figured out if they were real or if they're artifacts. And I would say that is the most important advice I would give me or anybody else, I think, starting out in science is just because you're going into lab to do a thing doesn't mean that's the only thing you're going to learn.
Well, those are all the questions that I have for today. Thank you so much for taking us through your journey as a scientist, Bruce. We hope that your words inspire students who hope to pursue a career in science. For those watching, if you're interested in the research at the Molecular Foundry, stay tuned for upcoming episodes of Foundry Fireside. Thank you so much for your time, Bruce.